Hello everyone and welcome to this presentation as part of the Wisconsin Science Festival. My name is Jonathan Mitchell and I'm a senior research scientist working at Promega. And today I'll be discussing with you some of the basics of virology with an emphasis on the SARS coronavirus 2. So to begin with, let's ask the question, what is a virus? Well, most of us would define a virus as being an infectious, microscopic agent that's capable of causing human disease. But what we're going to discuss today is what is a virus like at the molecular level. So viruses come in many different shapes and sizes. Here we're looking at a cartoon of a variety of different viruses, looking specifically at the viral particles, or virions. And so again, viruses vary widely. However, there are some common elements that are shared between different viruses, and we'll highlight some of these in the context of the SARS coronavirus 2, which is of course the causative agent of the current COVID-19 pandemic. So all viruses will have a genome that's comprised of either DNA or RNA. This viral genome is enclosed or encapsulated in a protein shell known as the capsid. So in the case of SARS coronavirus 2, we have an RNA genome coated with this nucleoprotein or N protein that protects the viral genome inside the particle. Outside of this then, some viruses have what's called a lipid membrane or envelope, which provides an extra layer on the particle. And then embedded within this membrane or this envelope are different viral proteins that are typically involved in the entry of the virus particle into its host cells. So again, in the context of SARS coronavirus 2, this is going to be mediated by the spike glycoprotein shown on the outside of the particle here. And of course, viruses have to enter a host cell because viruses can't multiply on their own. So viruses require a host cell in order to multiply. Okay, so let's talk then about how do viruses multiply. Well, shown here is a very general outline of the stages of virus multiplication. The example given here is for an influenza virus, but these stages are broadly true of virtually all viruses. And so first we have the entry steps, which are shown across the first three steps here, and we'll go into more detail on these later. Once the virus has entered the cell, it needs to replicate or go through a biosynthesis stage in which it creates more viral genome, more viral proteins. These genomes and proteins then have to come together in the form of new viral particles during the assembly stage. And then ultimately, these viral particles must be released from the infected cell in order to go on and infect more cells. So some important things to realize about virus multiplication is that each stage in this process is going to be composed of many smaller steps. And these stages and steps are going to be different for different viruses. Each of these stages and steps is also a potential target for us to develop antiviral drugs that can inhibit virus multiplication. And from the standpoint of a company like Promega, it's important to know that each of these stages of virus multiplication is a potential point at which we can measure, study, or assay the virus and its multiplication. So at Promega, we specialize in developing new technologies and assays for studying different biological processes by harnessing the power of bioluminescence. So bioluminescence is simply the emission of light by a living organism. Of course, the first example that probably comes to most of our minds would be a firefly. The genes that encode these bioluminescent proteins can be identified and isolated, such as the example of the firefly luciferase gene. Once these genes have been isolated, they can actually be inserted into the genomes of cells, bacteria, or even viruses. So here we can imagine inserting that luciferase gene into a viral genome and then generating a virus that we can then track using the light emitted by these bioluminescent reporters. And this can be used to study many different processes during virus multiplication. And so for the rest of this presentation, what I want to focus on are, again, the basic stages of virus multiplication with a focus on SARS-2. But I also want to highlight different applications of bioluminescent reporters for studying viruses, including SARS-2. So let's start with the first stage of virus multiplication then, which is virus entry. You can think of this as being the breaking and entering stage where the virus gets into the host cell. So entry is made up of 4 smaller steps, starting with the attachment of the virus particle to the host cell receptors on the outside of the cell. In the case of SARS-2, the virus actually uses the host ACE2 protein or ACE2 receptor for cellular entry, and this binding is mediated by the spike protein on the outside of the particle. Then comes the penetration stage where the virus particle actually enters into the host cell. And so again, in the case of SARS-2, this can occur either directly into the cell or into the cytoplasm, or the virus can be taken up by the process of endocytosis into these vesicles. Then once the virus has physically entered the cell, it needs to somehow fuse its viral membrane with these cellular membranes. And so again, in the case of SARS-2, this is mediated by that spike protein on the outside of the particle, which triggers the fusion of the viral envelope with cellular membranes. This is also driven in part by some of the host cell's own protease proteins. These are proteins that cleave other proteins. These happen to cleave the spike protein in a way that facilitates the fusion. And then once fusion is complete, the virus needs to release and uncoat its viral genome in order to set the stage for the next part, or the next step in multiplication, which is replication. However, before we go on to the replication stage, let's talk a little bit more about virus entry and how it's a very prime target for the development of antivirals, vaccines, and other drugs. So virions can be targeted separately from the host cell, which is one of the major advantages of targeting the entry step. Once the virus has gotten into the cell, it becomes much more difficult to target the virus without having effects on the host cell. And then each step in this virus entry process is a potential target for antiviral drugs. You can imagine drugs that block the entry step, the fusion step, and any of the other steps along this pathway. And there are many different types of therapies that can target virus entry. Probably the most classic type of therapy would be vaccines. So vaccines typically will elicit the development of neutralizing antibodies. These are antibodies that can coat the viral particle in a way that prevents it from entering the cell. You can also have immunotherapy, either using plasma from patients who have successfully encountered and cleared the virus infection, or you can have antibodies generated in a lab setting that can act much the same way as these neutralizing antibodies. And then there's, of course, other drugs that can target viral or host entry factors involved in this process. And so the majority of vaccines and therapies under development for SARS-2 now do target this entry step. And so bioluminescent reporter assays can be used to monitor this viral entry and to assess the potency of these different antiviral drugs. And so Promega is actively developing now multiple bioluminescent reporter assays to monitor the entry of SARS-2. So let's move then to the next stage of virus multiplication, which is the biosynthesis or replication stage. And you can think of this as the virus essentially hijacking the host cell in order to create more copies of itself. So there are 2 primary steps in this replication process. There's the expression of viral proteins, and then there's the replication of the viral genome itself. Now the specifics of this stage, like other stages, is going to be different from virus to virus. And for replication, these specific details are largely dictated by the nature of the viral genome. That is, viruses that have a DNA genome are going to replicate in different ways than viruses that have an RNA genome. But in both cases, replication is going to be driven by viral proteins and many hijacked cellular proteins. And so viruses will manipulate the cellular environment in a wide variety of ways in order to essentially convert cells into factories for virus replication. So here is an example of images taken directly from a coronavirus infected cell, where we see the virus has established these really unique replication complexes. These are like little mini factories where the virus is producing more viral RNA to be assembled into new viral particles. And so some more specifics then on SARS-2 replication. So SARS-2 has a positive sense single-stranded RNA genome, which means that this viral genome can be translated directly by host ribosomes, much the same as a messenger RNA. Coronaviruses have the longest known RNA genome amongst any viruses at about 30 kilobases. This is a very, very large genome for an RNA virus. And these genomes are somewhat unique in their ability to undergo RNA recombination, where different strands from different coronaviruses can actually exchange. And we think that this is an important driver of the coronavirus evolution process. And so just to highlight here, again, the schematic showing the genome of the virus, and then the long polyprotein that's made off of this genome. And there's some key proteins for the replication process that are highlighted here, starting with two different proteases, PL-Pro and 3CL-Pro, which are involved in cleaving this viral polyprotein up into the individual viral proteins. And these are under investigation now as potential drug targets. Another important enzyme in the replication process is this RNA-dependent RNA polymerase, which is the enzyme that copies the viral genome or creates more copies of the viral RNA genome. It's also a target for antivirals, including remdesivir, which has been approved. And of course, the other thing to mention is that there are many, many host proteins that are going to be involved in the replication process of any virus, including SARS-2. And so recent studies have predicted over 300 different interactions between SARS-2 proteins and human proteins. So there's a really complex interplay between the virus and its host cell, where the virus is able to essentially, again, hijack that host cell in order to create more copies of itself. So again, this is a process that we can study using the power of bioluminescent reporter genes. And again, these bioluminescent reporter genes can be incorporated or inserted directly into the viral genomes. And this allows us to rapidly and quantitatively measure virus replication or the inhibition thereof. So just as an example here, we're imagining a scenario where we've incorporated this bioluminescent reporter into the viral genome. We then add that virus to cells to initiate an infection. Once the cell's infected, the virus will express this bioluminescent reporter from its genome, and so we'll see an accumulation of bioluminescent signal that's proportional to the amount of virus replication that's occurring in the cell. And again, we can use this also as a system to test different antiviral drugs, where here you can imagine adding that antiviral drug. As you add more and more of that antiviral drug, you inhibit replication, which we can track, again, using bioluminescence. So we see this decline in bioluminescence with more antiviral drug, indicating the inhibition of viral replication. And so bioluminescent reporters can also be used to measure other processes that are important for the replication process, like different protein-protein interactions. And they can also be used to measure the activity or the inhibition of individual viral proteins or enzymes. And so again, these are all areas of interest at Promega where we've initiated multiple projects for looking at SARS-CoV-2 protein-protein interactions and protein activities using bioluminescent technologies. So moving to the next stage, then we have the assembly stage. So the virus has now entered the cell, it's replicated, it's got more copies of its genome and a bunch of viral protein. It needs to put those genomes and proteins together into a viral particle. So again, you can think of this as the virus building its getaway car in preparation to flee the infected cell. So viruses have to form new particles or new virions. And here we have three particular requirements in this process. We need to assemble that protein shell, that capsid, around the viral genome. We then have to package the viral genome inside the capsid. And we have to acquire a lipid membrane or envelope. And so again, the way SARS goes about this, or SARS-2, is that the particles will assemble on intracellular membranes. So it's the nucleocapsid or nucleoprotein, N protein, that recognizes and coats the viral RNA, as shown here. So here we have that viral genome coated with the N protein. Then another protein that's involved in this process would be the membrane or M protein, which acts as an important scaffold to pull together the different viral proteins required to form a particle. And as the virus forms, it actually will bud or push its way through host membranes in order to acquire an envelope. Again, that's shown here. These are electron micrographs from an infected cell. So you can see here at the beginning of the budding process, where the virus is beginning to push its way, or the particles pushing its way into this membrane. Here we've moved one step closer. We're about to pinch off the membrane. And then finally here, the process is complete, and you see this brand new SARS-2 particle forming inside the cell. Of course, once the particle's been formed, it needs to be released. So viruses must be released for the infection of new cells. And there are two different ways that a virus can typically go about this release process. One way is to simply destroy the infected cell outright, usually referred to as lysis. Or the virus can seek to essentially hitchhike its way on the host cell's export pathways. And this is the way that SARS-2 is thought to operate, where SARS-2 virions will traffic to the cell surface inside of Golgi vesicles. So again, once the viral particle has butted into these membranes and acquired its viral envelope, it can be transported inside these Golgi vesicles to the cell surface and be released through the process of exocytosis. And again, like other stages, there are applications here for different reporter assays and bioluminescent reporter assays. So the assembly process is again largely driven by protein-protein interactions, which can be measured using different bioluminescent assays. Virus release can be assayed using imaging techniques with virus particles that incorporate different reporter genes. Or it can be possible in some circumstances to attach reporter genes or reporter proteins, including bioluminescent reporters, directly to the viral particles as a means to track those particles. So in summary, what I hope I've shown you is that virus multiplication is a very complex and a very tightly orchestrated process. And the specifics are going to vary from virus to virus, but the general stages of multiplication hold true across all viruses. And each stage in this virus life cycle is going to be a potential target for drug development and also a potential point at which to measure the virus's multiplication. So bioluminescent reporter assays can be employed to study many different viral processes throughout this multiplication process. And Promega is developing bioluminescent assays to make virology research, we hope, safer, faster, and less expensive. Thank you, and best wishes.